Section 4.2 Glycolysis. By the end of this section, you will be able to explain how ATP is used by the cell as an energy source, and describe the overall result in terms of molecules produced of the breakdown of glucose by glycolysis. Even exergonic energy-releasing reactions require a small amount of activation energy to proceed. However, consider endergonic reactions, which require much more energy input because their products have more free energy than their reactants. Within the cell, where does energy to power such reactions come from? The answer lies with an energy-supplying molecule called adenosine triphosphate, or ATP. ATP is a small, relatively simple molecule, but within its bonds contains the potential for a quick burst of energy that can be harnessed to perform cellular work. This molecule can be thought of as the primary energy currency of cells, in the same way that money is the currency that people exchange for things they need. ATP is used to power the majority of energy-requiring cellular reactions. ATP in living systems. A living cell cannot store significant amounts of free energy. Excess free energy would result in an increase of heat in the cell, which would denature enzymes and other proteins and thus destroy the cell. Rather, a cell must be able to store energy safely and release it for use only as needed. Living cells accomplish this using ATP, which can be used to fill any energy need of the cell. How? It functions as a rechargeable battery. When ATP is broken down, usually by the removal of its terminal phosphate group, energy is released. This energy is used to do work by the cell, usually by the binding of the released phosphate to another molecule, thus activating it. For example, in the mechanical work of muscle contraction, ATP supplies energy to move the contractile muscle proteins. ATP structure and function. At the heart of ATP is a molecule of adenosine monophosphate, AMP, which is composed of an adenine molecule bonded to both a ribose molecule and a single phosphate group. Ribose is a 5-carbon sugar found in RNA, and AMP is one of the nucleotides in RNA. Ooh, let's try that sentence again. I think there's a typo there. Ribose is a 5-carbon sugar found in RNA and AMP, and is one of the nucleotides in RNA. And adenine is one of the nucleotides in RNA. Let's try that. The addition of a second phosphate group to this core molecule results in adenosine diphosphate, or ADP. The addition of a third phosphate group forms adenosine triphosphate, or ATP. Figure 4.12. The structure of ATP shows the basic components of a 2-ring adenine, 5-carbon ribose, and 3 phosphate groups. The addition of a phosphate group to a molecule requires a high amount of energy and results in a high energy bond. Phosphate groups are negatively charged and thus repel one another when they are arranged in series, as they are in ADP and ATP. This repulsion makes the ADP and ATP molecules inherently unstable. The release of 1 or 2 phosphate groups from ATP, a process called hydrolysis, releases energy. Glycolysis. You have read that nearly all of the energy used by living things comes to them in the bonds of the sugar, glucose. Glycolysis is the first step in the breakdown of glucose to extract energy for cell metabolism. Many living organisms carry out glycolysis as part of their metabolism. Glycolysis takes place in the cytoplasm of most prokaryotic and all eukaryotic cells. Glycolysis begins with the 6-carbon ring-shaped structure of a single glucose molecule and ends with 2 molecules of a 3-carbon sugar called pyruvate. Glycolysis consists of 2 distinct phases. In the first part of the glycolysis pathway, energy is used to make adjustments so that the 6-carbon sugar molecule can be split evenly into 2 3-carbon pyruvate molecules. In the second part of glycolysis, ATP and nicotinamide adenine dinucleotide, or NADH, are produced. If the cell cannot catabolize the pyruvate molecules further, it will harvest only two ATP molecules from one molecule of glucose. For example, mature mammalian red blood cells are only capable of glycolysis, which is their sole source of ATP. If glycolysis is interrupted, these cells would eventually die. Figure 4.13. In glycolysis, a glucose molecule is converted into two pyruvate molecules.